Hello, everyone. Welcome to our monthly Science Vault Protein Seminar. My name is Amy Huang, and I'm the Associate Director of Scientific Ecosystems at the Good Food Institute. For those of you who are here for the very first time, welcome to our community. The Good Food Institute is a nonprofit think tank, 100% driven by philanthropy, and our goal is to build a world where alternative proteins are no longer alternative. We see scientists and engineers like you as being critical to that mission. If you're looking to take a deeper dive into the science of plant-based fermentation and cultivated meat technologies, be sure to check out our open access resources, analyses, technical publications at gfi.org/science. I am so delighted to formally introduce Dr. Lutz Grossmann to the stage. Lutz has been an assistant professor in the food science department of UMass Amherst since 2021. He graduated with a PhD in food science from the University of Hohenheim in Germany. His research focuses on facilitating a sustainable food system transition by designing holistic approaches to increase the consumption of plant and microbial protein-rich foods. Lutz is especially interested in combining up and downstream processing technology with molecular, physicochemical, and engineering concepts to create food textures that are nutritious, sustainable, and tasty. Lutz is amazing, as I'm sure you will experience firsthand for yourself. In short order, we're delighted to have him as a GFI grantee and so honored that he's here with us today to share his research and his many insights about the field with you today. So during today's seminar, Lutz will explain why hydrogen fermentation stands out as a really unique technology to convert electricity into hydrogen, and then ultimately transform it into a rich edible protein biomass that might have unique advantages for alternative protein products. So with that, Lutz, welcome to our seminar today. I will turn it over to you to begin your presentation, and thanks again for being here.
Thank you very much, Amy, and welcome everyone. Well, first of all, I want to say it's great to see all of these initiatives from the Good Food Institute. It's always a pleasure to see how this expanded over the past few years. And it's great to be here. I will start sharing my screen. So today I want to talk about, well, I framed it electroproteins. It's basically single cells that are derived from hydrogen fermentation. And the hydrogen can be produced from renewable electricity. That's why I coined the term electroproteins. So here you can see our campus. So we are at the University of Massachusetts in Amherst, which is in Western Massachusetts. Western Massachusetts traditionally used to be an agricultural state many years ago, before it all moved to the Midwest. And now you can see there's not much agriculture for many reasons. One reason is that the soil is not perfect, a little bit too many rocks. It's quite hilly. Then there's a high population density. So there's not too much traditional agriculture in Massachusetts, except for a few individual products. But we have a very strong fermentation community, especially in Western Massachusetts. So we have a lot of craft breweries, we have a lot of kombucha breweries, but up to now, we don't have a lot of new food biotechnology approaches that deliver us proteins. And as you can imagine, it's much easier to set up a bioreactor in such a hilly place than a traditional agricultural field. And that's why fermentation approaches that do not rely on traditional agricultural land are especially promising for an area like this, where we have really well-educated people that come out of the many universities that are in Massachusetts. Plus, we have the technology that can deliver us cultivated fermented foods. So my lab is really focusing on alternative proteins. We span from plant proteins to fungi proteins, but also to bacterial proteins like the hydrogenotrophs that I will introduce today. And I want to start with a number. And this number is 79. So imagine you have a process, a protein production process that delivers you 79% of protein without doing any extraction, isolation, refinement process. So just right out of your production process, you try your product, and then in the dry matter, you have 79% of protein. And then imagine even further, this process is not based on any agricultural inputs. So there's no sugary feedstocks involved. You don't have to add any glucose. But the only feedstocks that you supply are a few gases and a few minerals and a nitrogen and phosphorus fertilizer. And it comes out of the bioproduction process having a protein content of 79%. So that's quite special and unusual, even for a single cell protein. And this is what we can achieve with hydrogenotrophs, with bacteria that grow on hydrogen. And this is a recent number from our lab. And if you go to literature, some other authors even reported higher numbers, so well above 80% in the cell dry matter. And in my lab, we're also quite optimistic that we can even pump up this number a little bit. So that's quite a unique process, first of all, because of the protein content, and then given the fact that it relies on completely different feedstocks than conventional agriculture, but also conventional fermentation bioproduction approaches that usually rely on glucose feedstocks to deliver the energy to the organism that we want to grow in the bioreactor. But for those who are not really familiar with the whole food supply chain, I want to step one step back and briefly talk about where does this idea come from and where does the motivation come from that we want to maybe look into different types of supply chains to deliver us food that do not rely on traditional agriculture. So we have been doing agriculture for the past 10 to 15,000 years, and we've been really successful with it. And it's a really, really good production system. So I'm not arguing against agriculture, but as a scientist, I'm always interested in what else could we do? Maybe we missed an opportunity somewhere to develop novel supply chains that might be more efficient, more sustainable, might deliver us really interesting nutrients. And specifically for me as a food scientist, I'm always interested in, maybe there's an ingredient somewhere that gives us really unique functionalities that we then can utilize to produce new and better food formulations that are then may deliver better nutritional profiles or are even more accepted by consumers. So, but if we look on the current production volumes of agriculture, we see that 15 crops, so those are the 15 crops that are listed here, provide 73% of agricultural products. So you can see most of the raw commodities that we produce is sugarcane and corn. So obviously those also go into biofuel production. So not every single harvested crop from sugarcane and corn goes into the food supply chain, but still it shows you that we are quite limited in terms of what we are actually producing and put on people's plates. And not even in terms of diversity of what we produce, but also in terms of agricultural land. So we are already at a stage where we utilize too much agricultural land, and we still need to produce more foods in the future. So then the question is, where can this agricultural land come from if we agree to that we don't want to expand any more land use and for agricultural land? So obviously we could think about we make current agriculture more efficient, but maybe we can also find new methods for producing food ingredients that require much less space and that can be maybe scaled by volume and not by land area. So when we look at the current food supply chain, they all rely, basically almost all foods that we consume on a daily basis rely on two inputs. So one is sunlight. So we need sun energy, or if you grow it in a greenhouse, can also be LED light. But most of the harvested crops, they require sunlight to produce energy through photosynthesis. So this is one main input of our food system, sunlight. The other one is agricultural land. So we need land to produce crops. And this is a huge, let's say, a huge burden in some areas. And it's also a big driver for land use change and all of these issues. So then when we look into what kind of energy goes into the food system, so basically all energy that goes into the food system is somehow sun energy. So either through photosynthesis, so this is the food primary production where we grow plants or algae, they use photosynthesis to produce sugars that are then consumed by other organisms. So this is really the basis of our food system, sunlight. But sun also provides a lot of other types of, let's call it auxiliary energy that also goes into the food system. So right now we require approximately 10 to 15 kilojoules of fossil fuels to produce 1 kilojoule of food energy. And the main reason for this is that many fertilizers are based on fossil fuels, all the energy that goes into driving agricultural motor vehicles, for example. So there's a lot of fossil fuel that actually goes into producing foods. And fossil fuel is also just transformed sunlight. This is also algae and zooplankton that lived like millions of years ago and then was transformed into oil and natural gas that we then use today to produce fertilizers and to produce foods and transform these foods into other or different kinds of products. And then even also renewable energy relies heavily on sunlight. Wind energy relies on different temperatures within our atmosphere that then partly drives winds. Then obviously photovoltaic also depends on sunlight. So basically our whole food system is based on the energy input from the sun. So why I'm telling you all of this? I'm telling you all of this because I want to make a little bit thought experiment about what could we do, what kind of food could we harvest if there's no sunlight anymore. So based on my previous discussion, so there's not a lot. And especially when we look at the current supply chain, there's almost no food that we could produce without the energy input of sunlight. But just as a thought experiment to think a little bit out of the box, maybe there are also other ways to produce edible, let's call it biomass, that does not rely traditionally on the input of sunlight. And if we think a little bit out of the box, well, yes, there is a way that organisms thrive without the input of sunlight. And for this, we have to go a little bit into microbiology. So here you can see on the right side, this is like the ocean floor. And you can see a deep sea vent. They're also sometimes called black smokers. And these deep sea vents, they are generated through usually volcanic activity. So when there's a lot of volcanic activity, there are like superheated fluids that then basically open cracks in the deep sea floor. They flush out of the Earth's crust, and they carry a lot of fluids, gases, and minerals. And because they carry a lot of gases, these gases are often hydrogen, methane, hydrogen sulfide. And close to these plague smokers, we often find lots of microbial, but also animal activity. So in this example here, you can see that close to these vents and within these deep sea vents, there's hydrogen sulfide that emerges. And there's a specific set of bacteria that can utilize the hydrogen sulfide. These are called sulfur bacteria. So they use the hydrogen sulfide as their energy source. So similar to we use glucose as our energy source, those use hydrogen sulfide as the energy source. And they use this as an energy source, they multiply, and they live in symbiosis with mussel and snails that then feed off these kinds of bacteria. And when a bacteria doesn't use organic or compounds or sunlight for their energy, we usually call this chemosynthesis. So this is a process where inorganic compounds are used as an energy source. And this is usually not, this metabolism does not rely on actual input of sun energy. Okay, so what kind of bacteria are there that use this kind of metabolism? And there are all different kinds of bacteria that can utilize inorganic compounds. As I said, it's like hydrogen, methane, sulfur, all different kinds of compounds that can be used by all different kinds of sets of bacteria. So here are a few examples. For example, nitrate bacteria, iron bacteria, and also sulfur bacteria that we just talked about. But there's also a really specific set and a really interesting set of bacteria that are called hydrogen oxidizing bacteria or hydrogenotrophs. And these can use molecular hydrogen as their energy source. They're also called Knallgas bacteria because basically they carry out the Knallgas reaction in their cells, and you will see this provides a lot of energy. And these bacteria mastered using this reaction to produce energy, similar to we use glucose. Plants, they use sun energy, and those bacteria use hydrogen as their energy source. And the nice thing is, hydrogen can be produced from water and electricity in a renewable fashion. So it's a really interesting feedstock to develop a novel supply chain that is not based on all of the traditional inputs that we commonly use to produce edible biomass. So these bacteria, so where they come from and how do you get the idea of utilizing those? So again, we have to go to the deep sea ocean floor. And here again, you can see one of those black smokers, these deep sea vents. And within these deep sea vents, there's a lot of hydrogen release. And the hydrogen can come directly from magma degassing. So then it just comes out as pure hydrogen, or and now it gets a little bit of geology. So I'm not a geologist, so I hope I explained this correctly. But when different types of rocks transform under high pressure, high temperature, there's also a hydrogen production. And this is called serpentinization. So you have this olivine rock, which is a iron-rich rock, and it gets transformed under high pressure, high temperatures into serpentinite. So this is this rock over here. And during this process, the iron basically, takes all of the oxygen from water molecules and hydrogen is released during this process. And this happens naturally on the ocean floor. And then you have as well pure hydrogen released into the ocean. And then you can see in close proximity that there are these hydrogen oxidizing bacteria living in close proximity or even within these deep sea vents. And well, scientists, they're always eager to discover something new. So people took samples from these deep sea vents and found, so no one expected to find any living organisms there because it's really adverse conditions. But people actually found those hydrogen oxidizing bacteria living down there and then explored their metabolism. And it turned out that they can feed off pure hydrogen gas. And from there, a lot of research interest evolved. And from there, we figured out a few species that are actually edible. And two of these species that are currently especially of interest is Xanthobacter, which has been especially investigated by the research organization VTT from Finland. And then there is the Cupriavidus necator, which is also a hydrogen oxidizing bacteria and also heavily researched. Both of them are not yet food approved, but the Xanthobacter, for example, has, I think, preliminary approval in Singapore, for example. And then the first human and animal studies, they're really promising that food approval will be only a matter of time. So those are two species. And in my lab, we mainly work with the Cupriavidus species. And so how does this actually work? So I already said that this is the Knallgas reaction. And you can see here, it's basically a bubble foam produced with hydrogen. So within the bubbles, you have hydrogen. Then if you put in the activation energy, it burns up. So this is the typical Knallgas reaction. And you can see that's a lot of energy. So when you mix hydrogen, oxygen, it forms water. And this reaction releases a lot of energy. So it's 286 kilojoule per mole. So if you have 1 liter of hydrogen at ambient pressure, it has 2.6 kilocalories. If you have a hydrogen tank in your lab, so a regular 300 size tank, it has around 20,000 kilocalories. Okay, so this energy can be utilized by the cell for cell growth and for their cell metabolism. But how does it actually work in terms of biochemistry? So it's basically a half-finished photosynthesis reaction. So in photosynthesis, you have the first step, the light reaction, and then you have the dark reaction, which is the Kelvin cycle. These bacteria, they only use the Kelvin cycle to produce these carbon elements that are then used for producing sugars that are then either be stored or used for the metabolism. So what the cell does, you can see here, is H2, the hydrogen. This is like the outside of the cell. It then goes into the cell, and then the cell has a few hydrogenases. So the hydrogenases, they split up the hydrogen in protons and electrons. And then instead of releasing all of the energy, as we just saw in the video, all at once, it just goes through the typical electron transport chain, slowly brings all of the electrons from a high energy to a low energy input. And this energy is used to produce the typical proton gradient. And then this proton gradient is used to produce ATP. And then the ATP is used to produce or sequester CO2 and produce the C free carbon compounds that are then used in the metabolism or to produce sugars and just for regular metabolic activity. So you can see it's basically a half-finished photosynthesis reaction with only the Kelvin cycle without the light-dependent reaction. And the hydrogenase is like the main compound in the cell that allows the cell to utilize the hydrogen by splitting up the hydrogen in protons and electrons. So this is then basically the mechanism on how the cell produces the biomass. But the obvious question here is, where does the hydrogen come from? And we have currently, well, so people always come up with fancy colors for different types of hydrogens. So I think now there are even more, but we have 3, or let's say 4 main types of hydrogen. The main hydrogen that we currently use is gray hydrogen. It's more than 90% of the hydrogen that we get and use at the moment comes from gray, or is gray hydrogen, which is produced from natural gas. So there we take natural gas, which is mainly methane, we split off the hydrogen. During this process, we release lots of CO2 and we utilize non-renewable fossil fuels. So the gray hydrogen, it's very effective and a cheap way of producing hydrogen at the moment, but it's not really sustainable and it's not renewable. So then there's blue hydrogen, which also uses fossil fuels, but we take the CO2 that develops during the hydrogen production, and then we store it underground, or we transform it into other types of products. So we don't release the CO2 in the atmosphere. So this then is associated with a little bit less CO2 emissions. But what we want to have is green hydrogen. So green hydrogen is produced from electrolysis. So we take water, split it with electricity, and then we can harvest the hydrogen. And we can drive this process with renewable energy. The only disadvantage is that at the moment, this process is still way more expensive because natural gas, which is commonly used to produce gray hydrogen, is still much cheaper than using electricity and water to produce green hydrogen. But as we scale up this technology, and there's a big interest in scaling up green hydrogen production for various reasons, for example, for green ammonia production that then allows us to have green fertilizers. The Department of Energy currently aims or has this milestone of reaching $1 per kilogram of green hydrogen. And if we would become, or if we could get the green hydrogen price below, so around $1 per kilogram, then it would be a really, really feasible process. But overall, at the moment, we can use green hydrogen to produce this biomass based on hydrogen. And obviously, we can also do this in the lab because we can just produce really easily in the lab green hydrogen, just using electricity and an electrolyzer, and bubble this into our bioreactor. And that's exactly what we are doing. Another thing that just is up and coming is so-called gold hydrogen. So gold hydrogen is basically hydrogen from natural resources. At the moment, it's quite interesting that at the moment, we don't really know how much natural hydrogen resources there are. But it's estimated that there could be vast amounts of natural hydrogen sources that we could actually harvest. And these are also produced through similar principles that I explained before. So there's again this process of serpentinization, which is this rock transformation where hydrogen is released. But then there's also radiolysis, where radioactive material splits water, then hydrogen is released. And there can also be like direct vents basically in the earth crust where hydrogen is released. And at the moment, there are huge exploration activities globally to find natural hydrogen resources that may basically solve the problem of hydrogen availability. But this is really something that is still up to, well, we have to actually find those. But if we find those, it might be a really, really good opportunity. It's a little bit of a gold rush at the moment to find these natural hydrogen resources. But so how do we actually do this? So how does the fermentation process look like? And how can we design the overall cultivation process? So here you can see a rough sketch on how it works. So we use renewable electricity. We use this electricity to drive our electrolyzer, our hydrogen generator. We then use the hydrogen. Potentially, you can also use the oxygen, but let's focus on hydrogen. We can use the hydrogen coming from this process. We bubble it into our bioreactor. Then we need to feed as well CO2. So this either use direct air capture, or in the lab, more convenient, you just use a CO2 cylinder, bubble it into your bioreactor together with the hydrogen, and then you also need to bubble in oxygen. So you have the three gases, oxygen, hydrogen, CO2, that you bubble into your bioreactor. Then you need a nitrogen source. So this is can be ammonia or urea or any other ammonium compound. This can either produce through electrodialysis of water, or it can also be green ammonia if we scale up again the hydrogen production to actually be able to produce lots amounts of green ammonia. You feed this, and then you need to feed a few minerals. So you have your three gases, ammonia, phosphorus, and a few minerals that you feed. This is all that the organism needs. You let it grow similar as you would do with a regular bioproduction cultivation process. You harvest the cells, do your downstream processing, and then we hopefully can produce these kinds of food ingredients that then can be transformed into, for example, meat alternatives, dairy alternatives, or any other types of foods that we can think of. But obviously, if you work with hydrogen, you have to think about that it's a flammable gas. We saw this in the short video, that hydrogen is a flammable gas, so you have to be diligent in your reactor design and how you actually feed the gases and at what concentrations you feed the gases. So I will talk on the next slide a little bit on how we designed our bioreactor to make sure that we don't create any hazardous environment, that we don't create any flammable environment. But in general, we have a lower flammability limit of hydrogen, which is 4%. So if you have oxygen, you put in hydrogen into the oxygen atmosphere, then as soon as the hydrogen concentration reaches 4%, you get a flammable atmosphere. The other way around, if you have like 100% hydrogen and you put in oxygen in, then when you have 94% hydrogen, 6% oxygen, then you create a flammable environment. So one obvious solution to not creating a flammable environment is to just use oxygen levels below 6%. If you have oxygen levels below 6%, basically there's no way that the gases will ignite. And that's exactly one strategy that we perform in the lab beyond other safety measures. And here you can see our setup. So one safety measure that we do is we go below the flammability limit. So we never have more than 6% of oxygen in our system. You could create all of this in a way that you could also cultivate above the flammability limit, but then you would need to add more safety measures. Then you would need to have like an explosive-proof environment. And in our setup, we don't have this, which is not a problem because we designed it in a way that we are not creating any flammable mixtures. And this is also then easier transferable to other labs because many labs, they don't have an explosion-proof environment where they can work, where they can do such cultivations. So this technique is easily transferable to other kinds of labs. So how do we ensure that there's no flammable environment? So in two ways. So first of all, you can see our reactor is in a fume hood. So meaning if there's any leak, the hydrogen will be immediately diluted below the flammability limit. Second, what we do is we have two separate spargers. So one hydrogen sparger and one O2 and CO2 spalcher. So in this way, the hydrogen and oxygen, they only mix in our media, and in the media you can't have any ignition. So that's another way to minimize any flammability risk. Then when the gas reaches the headspace, there is a potential for, let's say, creation of flammable mixture. We prevent this by, first of all, diluting the headspace with nitrogen gas. Then we have an air-driven motor, so there's no ignition source. And we don't, as you can see here, our reactor is completely glass and Teflon lid, so there's no electrostatic discharge. So even if there's a flammable mixture, there's basically no way that there's a source of ignition that could actually ignite the mixture. And then if we went it in the fume hood, it immediately diluted. But we already diluted in the headspace, so in this way, all of our exhaust gases, they are already below the flammability limit. So yeah, that's how we do it. So basically creating a low oxygen atmosphere, mixing the gases separately, and as soon as they had the headspace, we dilute it, and then we vent it into our fume hood. So in this way, we can ensure that there's no real flammability risk. Okay, so how does it look like in terms of cultivation conditions? So here's an example of biomass yield reached. So here you can see they used a 1 liter reactor here in this study. They reached 91.3 grams per liter, which is really, really high biomass yield. And the protein content is 75%. So this process actually gives you like really high biomass yields, which is also not typical, not usual for many of these fermentation approaches. Oftentimes the biomass yield is much, much lower. Then what is the actual content? So here you can see I already said the protein content can be really high. So here in this study, the reported protein content was around 75% in the cell dry matter. And you can see the amino acid composition here. So as of now, we don't really know how is the digestibility, what's the PDCAS, DIAS value, all of these protein quality parameters. But just from looking at the amino acid composition, you see really high lysine content, so it might be a perfect fit to mix this kind of ingredient, for example, with cereals, because cereal proteins are often deficient in lysine. So there might be a mutual benefit of mixing these two ingredients. But overall, really, really high protein content, high biomass yields, so a really promising cultivation approach. But then the obvious question is it actually sustainable? So how does the life cycle assessment look like? And as you can imagine, this highly depends on what kind of electricity you're using. If you're using just a regular electricity mix, where most of the electricity is generated from fossil fuels, then your carbon footprint won't be that beneficial. So here you can see life cycle assessment. So two things highlight. So the land use is quite low because you only need the land to produce renewable energy, and then your bioreactor. So the land use is low, and the carbon footprint, if you utilize electricity that is produced from solar energy and wind energy, is also comparably low, especially if you compare it to animal foods. And these are just the first studies. So we haven't really optimized the whole process. So I would expect that these numbers probably even go down in the future. But you have to ensure that the electricity you're using for the process is based on renewables. Then the question, is it efficient? So how much food do we actually get out per area of land? And when you look at the energy balance, so let's assume you get like 100 kilowatt hours of sunlight. Then we lose around 80%. So we can make around, from 100 kilowatt hours, we can make around 20 kilowatt hours of electricity. We lose a little bit again through the grid. Then current electrolyzers have an efficiency of like up to 80%. So 80% of the electricity that goes in is transformed into hydrogen gas. So theoretically, from 100 kilowatt hours, we have hydrogen gas with an energy of 13 kilowatt hours that we then can use in our bioprocess. But then obviously at the moment, we don't really know how much of this gas is actually used by the organism. Also depends on the process, whether we recycle the off gases or we just let it vent. Then how much energy do we need to run the process? So the stirrers, heating, cooling, etc. So the question is, how much biomass do we actually get out from 100 kilowatt hours? It's an open question. But if you compare it to photosynthesis, which has an efficiency of around 1%, so from 100 kilowatt hours, you get 1 kilowatt hour of food energy. There might be an opportunity here to have higher efficiencies compared to the regular agricultural production stream. And this also has been highlighted in studies. So here you see a comparison between how many people or how many protein can you supply per hectare. So here, 1 hectare for a hydrogenotrophic production line. So here you have photovoltaic-driven electricity production, which requires land. You have land that you need for your bioreactors. Then here in this study, they also assumed that there's direct air capture to produce CO2. And with 1 hectare in this hydrogenotrophic production process, you can feed around, or you can provide proteins for around 520 people, assuming that they consume around 80 grams of protein per day. If you compare this to a, let's say, traditional fermentation process where you have to grow sugar on a field, then you harvest the sugar, extract the sugar, and then feed it into your bioreactor, you can feed around 90 people. And with a traditional soybean field, you can feed around 40 people. So this is a little bit hypothetical because it's based on just assumptions, and no one ever has so far did this kind of production, but it shows that there's a potential for highly efficient protein production using such gas cultivation technologies. And I worked a lot with microalgae in my research. And when you compare the two technologies, they have almost same arguments, saying, okay, we don't need traditional agricultural land. Both technologies can thrive on renewable energy or just sunlight, and they don't compete like with traditional agriculture. So a disadvantage of microalgae production is that it's limited by the surface area, because you need huge surface areas to ensure that each algae gets enough sunlight so that you get a really efficient production. So all scaling is based on surface area, which is a limiting factor, versus a gas cultivation process, what you can see on the right, a chemotrophic organism. We can scale it by volume because we can actually mix the gases in the bioreactor, and in this way scale by volume, not by surface area, which can be an efficient way of scaling such production technologies. Okay, but obviously there are many challenges ahead. That's why I'm working on this academic research. I'm always interested in solving problems. It's not an established technology yet. It's really still in the early stages. And one challenge that we face if we want to scale this is the hydrogen availability, and especially the green hydrogen availability. So we can produce gray hydrogen, but as I showed you, it's not really sustainable. So if we want to scale this process, we need much more green hydrogen. And at the moment, it's only around 2% of hydrogen that is produced through electrolysis. And there are estimates, if we take or if we transform all of the hydrogen production that is already established, so all of the gray hydrogen production, and we produce it through water electrolysis, then this would require more electricity than the European Union currently uses. So this means we need not only ramp up green hydrogen production, but we also need to ramp up renewable electricity production to make this worthwhile. And when we think about what other things we also want to electrify, this might be a challenge in the future to say, okay, where do we set our priorities? Who gets all the new renewable produced electricity? So this will be a challenge, where to get the green hydrogen from if we want to scale this up. And then from a food science perspective, the question is, it sounds like a really nice technology, sustainable, doesn't need a lot of land, really new feedstocks that do not require any agricultural input. Well, the question is, what consumers will ask, what is it? Can I eat this? And then what we as food scientists and food industry will ask is, how functional is it? How can we produce foods out of it that then consumers will accept? Is it nutritious? Does it provide like all the amino acids? How digestible is it? And all of these questions, they haven't been answered yet. So that's also then still requires a lot of research to answer these questions in terms of what kinds of ingredients can we produce and how nutritious is it. And what's holding us back at the moment is that we don't have enough of the raw material. That's why we set up the bioreactor in my lab, because we want to produce the raw material to study it and understand it. But at the moment, we just don't have enough raw material to study this, all of these properties. Because most of the research that has been done was in small quantities like milligrams, but for food science studies, usually you need a few 100 grams to run your experiments and understand how you can actually utilize and use the raw material. So that's a challenge. Then electricity prices. So you can imagine that this process is basically almost based 100% on electricity. So if electricity price change, this will significantly affect the price of this production technology. And here you can see the electricity price in New England over the past 20 years. You can see it actually fluctuates a lot. So if your company wants to produce hydrogen trophic biomass, you have to account for fluctuating electricity prices, because this will affect the price of your final product. But maybe in the future, if we have like abundant renewable electricity, maybe this won't be a problem anymore. Or let's say, I would really hope for this. But this is something to account for. And then lastly, the obvious question is, can I eat this? As I mentioned, there in Singapore, Xantobacter got food approval, but there's no food approval yet for Cupria vidus. But this study was published, I think, end of last year. So this is a toxicology study with Cupria vidus. And the conclusion of the study was that it's safe, or that they didn't observe any adverse effects up to 3 grams of body weight per day. So this also is very promising in terms of that we see actual foods or food approval soon for these hydrogen-trophic organisms, and then have a completely novel way of producing foods that has never been done or seen before, basically. And with this, I want to conclude. I hope I could give you a few insights into this kind of new technology and new supply chain to produce proteins. So my vision is really, you have the hydrogen and you transform the hydrogen into a kind of meat alternative product. We are not there yet, but I hope within the next few years, we will be able to show that this is a viable technology that also delivers functional and nutritious foods. So to summarize, the advantages are really the low land use, low emissions, little need for fertilizers. I didn't mention this, but every bioreactor, we don't have problems with runoffs. So you only need little fertilizer and no or only little pesticides. There are limitations like the electricity requirements. Big question also how to do the reactor scale up in a safe manner. The food applications, and last but not least, will consumers actually accept this kind of high-tech type of food that is not that is very, very different from what they are used to. But overall, as always, I always highlight more research is needed, really new technology, really exciting. If you want to learn more about, we published a review paper last year. If you scan the QR code, it will lead you to the paper. If you don't have access, shoot me an email. I'm happy to share the manuscript. Then I want to thank my PhD student, Carlos, who does all the heavy lifting and carrying out the research and heavy lifting in actually because you always have to put the reactor in the fume hood. So I want to thank him for all of his dedication and work. And then the Foundation for Food and Agriculture Research, which funds this research that helps us a lot. And then thanks to Good Food Institute for giving me this opportunity to share all of this with you today. I think now it's time for a Q&A. So yeah, happy to answer your questions. And yeah, if you want to get in touch with me, write me an email or connect with me on LinkedIn. My email, I didn't put it there, but it's lkgrossmann@umass.edu. Thank you.
Thank you so much, Lutz. This was a fantastic presentation. I learned so much over the course of the process, and given all the questions that are coming through, I'm guessing many of our attendees did as well. For those of you in the audience who have not yet gotten the chance to ask your question, please be sure to drop it in the Q&A section of your Zoom bar. That's where we're processing things through. I'm seeing some questions come through the chat, so if you can redirect them to the Q&A section, that would help us prioritize the queue. Thank you so much. Again, for this presentation, Lutz, one of the threads of questions that we have throughout is around the life cycle analysis of these technologies. One question, if we can kind of revisit your methodologies for the life cycle analysis, is just why the carbon footprint is still so high, even in the scenario where renewable energy is used, given that your hydrogen oxidizing bacteria is consuming CO2. Can you kind of illuminate a little bit or reiterate what the main greenhouse gas sources or carbon footprint seems to reside?
Yeah, that's a good question. So it can be different things. So it depends a little bit on how you set up your process. So first question is, where does the CO2 come from? So if you haven't set up a direct air capture, then you have to use basically CO2 that comes from a compressed cylinder. And then if you don't recycle your gases, you have to release it into the environment. So this can be one cause of greenhouse gas emissions. And there again, if we advance our production, our cultivation technology, maybe we don't lose any gases anymore in the future. Another source is the nitrogen fertilizer. So at the moment, it's really difficult to obtain sustainable nitrogen fertilizer because basically all of the nitrogen fertilizer that we get is produced from natural gas, and this has a carbon footprint. And then also renewable energy, oftentimes still, it depends a little bit on the life cycle, but while the production of the renewable electricity has usually zero carbon emissions, but you still need to produce and manufacture, for example, your turbines, your solar cells and all of this. And this also creates emissions that you have to account for. So it depends a little bit. So where do you start your life cycle assessment? Where do you end it? Where does all of your ingredients come from? While maybe you can drive like the hydrogen production and also your process 100% with renewable energy. Yeah, then the question is, where does the CO2 come from? Do you vent it again into the atmosphere or do you recycle it? Because you always have to imagine when you bubble in gases through a bioreactor, like 99 point whatever percent is not utilized by the organism, but just ends up in the headspace. So, and then it really depends on what do you do with the exhaust gases.
Super helpful. Thank you. It makes sense that the kind of sector boundaries are really, really important for
Yes.
what ends up being captured in these analyses. Follow-up question to that. You mentioned direct air capture as a method throughout. Can you share a little bit more about how CO2 direct air capture works?
Okay. So I'm not so I'm a food scientist by training. So I can't go into that deep into how it works. But basically what you do is you take air, you have potassium hydroxide. If you bubble air through potassium hydroxide, you can basically extract the CO2 from the air, and then you take the basically storage CO2 in the potassium hydroxide and transform it. I guess they use some kind of calcium mineral. Then you release again the CO2, the highly concentrated CO2. So CO2 concentration in air is 400 ppm at the moment, so it's not really high. So you need to concentrate it first. And this is done through the potassium hydroxide. Then you, I think, transform it into some kind of calcium mineral. Then you can release it from there, and then you have like highly concentrated CO2. Then you can either use this CO2 or what a lot of people propose, you store it underground. So yeah, that's my understanding how it works. But as said, there's no guarantee that this is actually true.
You are multi-talented and represent all of the disciplines across the scientific sphere. No, that was great. Thank you so much, Lutz. A follow-up to your comment about hydrogen sourcing being an important consideration here. There's a question around whether the CO2 comes directly off of a flue stack or if it needs to be purified, concentrated, kind of containerized first prior to usage.
So when you think about setting up a process, and from a process engineering standpoint, you want to control the concentration of gases because this will impact your yields. So and also in terms of, especially in this kind of process, in terms of process safety, you need to ensure that you have an optimum gas mixture for, as I said, safety, but also to achieve optimum growth and protein yields. So yes, one option is to use flue gases from another type of process if it's CO2 rich, but then you need to know how much CO2 is in there so that you know what is the ratio you need to mix in your other gases, so the hydrogen and oxygen. But it's definitely an option to use exhaust gases from another type of process as a CO2 source. And the purer it is, the better.
Super helpful. Thank you. There's a question here around the use of gene editing as an alternate method to producing pure proteins. So instead of using these bacteria as your end product, have you considered genetic engineering that might be able to produce specific pure proteins like how E. coli has been used as a platform, as an expression platform? Would that help us get around some of these challenges?
I think absolutely. And also, what I briefly showed is that the hydrogenase is like a really key enzyme in the organism to basically harvest and split the hydrogen. So I could, for example, envision to use metabolic engineering, genetic modifications, to overexpress this enzyme so that you have lots of hydrogenases within the cell, and in this way then increase the hydrogen utilization in the cell. So I think even within this process, using metabolic engineering probably could help a lot to make the process even more efficient. But then so that's like that's I would say scientific point of view. But then you have to obviously think about, okay, yeah, it's getting more difficult with getting food approval for this. And the question, as always, is consumer acceptance. So then you would definitely need to label it as, well, in the US, you have to label it now as bioengineered food. In Europe, you would actually have to label it as GMO. So then there's the question, would the consumer accept it? But from a pure scientific standpoint and efficiency standpoint, I think there's a huge potential in using metabolic engineering to make this process even more efficient.
Thank you. There's a related question here about what feels like all this extra technology compared to your standard classic precision fermentation methodologies. People are kind of curious if you feel like this technology will be scalable enough in terms of capital and innovation that's needed, or might it fall into some of the pitfalls that you also kind of mentioned there, that vertical farming has fallen into in terms of using too much technology and infrastructure to try and solve this problem. So very curious to hear, you commented throughout on the massive scalable potential of this technology, but curious if you see any immediate pitfalls in that same realm.
Yeah, very valid question. And I think it comes down to, so we can build the reactors. I think the reactor technology is actually not that complicated. It's not a very specific type of reactor. We also over-engineered our reactor a little bit in the lab. We are Germans, so we like to over-engineer things. It can also be done a little bit easier. But the reactor technology is quite common technology already. It's not that we couldn't scale the reactor itself. I think the main challenge will be the green hydrogen. But there I'm actually quite optimistic because there's globally so much incentive in making green hydrogen and bringing the price of green hydrogen down within the next decades. So in terms of scaling the green hydrogen production, I'm actually quite optimistic that this will be achievable in the next, not tomorrow, not next year, but within the next 10, 20 years, that we will see that green hydrogen will be really, really cheap. And then if we continue to making this process more efficient, working again further on making the bioreactors more efficient, starting many more bioreactor companies, that's really important. For example, when we started to look into finding a company that could build us a bioreactor, like we call a lot of companies, bioreactor companies, basically they hang up on us because they said, oh yeah, we don't have any capabilities of utilizing hydrogen as a gas feedstock. But I think now with that we develop more and more of these technologies and gas fermentation technologies, in the future the companies will learn how to deal with this kind of gas and to ensure that it's safe to use. Similar to what we learned with natural gas, we know how to handle it. We have cooking stoves at home that work with natural gas, which is like a highly flammable gas. So why shouldn't we be able to learn how to handle hydrogen in a safe manner? So yeah, I think it's we need more innovation in the bioreactor space, but also the green hydrogen price has to come down. So it won't be a technology that we can use tomorrow, but hopefully in the next few years.
Thank you. Yeah, so many great comments there. And I think that this is a really strong example of how we'll be riding the momentum and leveraging advancements in parallel sectors to make these types of applications a reality. Back to bioreactor design for a moment. There's a question from an audience member about whether you utilized computational methods or simulation or modeling to optimize your bioreactor design at any point. That seems to be a really impactful and underexplored intersection.
Yes, coming up. So this is what we are currently doing.
Great, short and sweet. That's fantastic. One question that remains here is around whether you can comment on the quality of the protein that's produced by this method, maybe specifically focused on amino acid composition, or other important profiles.
So not yet, unfortunately. So we know the amino acid composition, but we know that having the composition doesn't really tell us about digestibility. So it's like two different things. So based on the amino acid composition, we can draw some preliminary conclusions. But if we really want to understand the protein quality, we need to know the digestibility of the, well, either of the bacteria itself, or if you produce protein extracts from the bacteria, then we need to study what is the actual protein or amino acid availability within these ingredients. And because so far the availability of this ingredient is really limited, so no one has done it yet. Maybe someone somewhere is doing it at the moment, I hope so. But we don't have these numbers at the moment. So it's difficult to draw like a conclusion. But I hope it will be of a high protein quality in the end. And then this is like one species that I showed. So we don't know what is the amino acid composition. Maybe of other species, maybe it's better. Maybe the digestibility is better. The amino acid availability might be better in other species. So yeah, there's still, I hope everyone will get into the space because there's still so many open questions that we have to solve.
That's an awesome call to action. There's a question here, borrowing on your comment around species. What types of cultures did you consider in going into this research design? Did you consider mixed cultures or only pure cultures like C. negator?
Yeah, so we thought about having mixed cultures, but right now we only rely on one culture. And we also wanted to work with an organism that is promising for food approval, and we're also startups are working with that also then want to bring this to the actual food market. So that's why we decided we go with Cupria vidus. And a big advantage of the technology that I also didn't mention is because we don't have sugars in there, the risk of cross-contamination is pretty low. So if you have, for example, a fungi fermentation process, very high risk for contamination, especially with bacteria. But in this process, because not many organisms can grow on gases, the risk of cross-contamination is much lower than in other types of cultivation processes that rely on sugar feedstocks. But yeah, at the moment, we only use single species culture. But there's a good opportunity to use to do multi-species cultivations. You could envision, for example, that you grow a organism that fixes nitrogen for you and then lives in symbiosis with a hydrogen oxidizing bacteria. So there are also much more opportunities to be investigated here.
Yeah, that benefit of low cross-contamination risk seems like a hugely important one for its application in the food industry, especially at larger scales. So very, very cool. I didn't thought about that. There's a question here about in situ hydrogen production. So basically performing the electrolysis directly in the bioreactor. That also gets rid of some of those hydrogen issues, but might um, result in a hit to efficiency. Have you completely taken it off the table? How are you thinking currently about direct in bioreactor electrolysis?
Yeah, it's an option. So we haven't really explored this opportunity, but not for a specific reason. So to me, I can't see why this wouldn't be an option to produce the hydrogen as well within the bioreactor, eliminate some problems with hydrogen transportation into the bioreactor, and you could then also directly have the oxygen in your bioreactor. So it's definitely an option. For us, it was just a little bit easier, I would say, to have the hydrogen generation outside of the bioreactor. So there's no particular reason why we didn't put it in. For us, it was just from a design standpoint, it was a little bit easier to do it like this.
Great, thanks for continuing to illuminate your research study design. A couple last questions here before we wrap up. One question is around what trace nutrients you ended up using throughout the process and whether you were able to source them as food grade materials.
Yeah, that's a good question. So, so there's the trace nutrient media, and well, if the person, we are happy to share it and what we exactly use. But yes, there might be a challenge of currently utilizing this as food grade materials, as food grade minerals. But I don't see why this shouldn't be feasible in the future to source these food grade minerals, because well, in the lab usually you use your chemical vendors to supply you these minerals, but I don't see why you shouldn't be able to find a distributor that is able to certify that you can use these minerals in foods. And then there's also so usually you use kind of standardized media, usually from literature databases, but for most species, these media haven't been optimized. So we don't know exactly, maybe we can get rid of a few of those minerals because our specific organism maybe doesn't even need it. So that's also like optimizing the cultivation process that hasn't been done yet.
Fantastic. I am dropping your email in the chat
Yes.
for any who are interested in collaborating on any array of topics. It seems like a very underexplored space, and I'm so glad you're beginning to chart the path forward here. Hopefully, there will be many multi-institutional collaborations to come. Um, I want to shift gears just a little bit in our final few minutes. I know that in addition to doing really impactful research, you also have some pretty exciting educational initiatives in store. Do you want to share a little bit about what you're doing on the education front?
Education front. So yes. So well, several things I would say. So we are establishing novel courses at UMass Amherst that specifically focus on sustainable food production. So there we will talk or teach our students sustainable processing, sustainable ingredients, how to formulate foods with novel sustainable ingredients. They learn about, for example, extrusion processing, plant proteins, plant lipids. I will also introduce like these novel technologies, cultured meat, gas fermentation. So we currently establish like a more focus at UMass Amherst to educate our students more on sustainable food technology. Then our students, they also apply for the GFI student chapter. So that's really good. And we are currently discussing and ramping up a sustainable food science master's degree. So that might also come in the future. So I think to me, it's like really important that students are aware of the impact that the food science community has on the environment. So I think that's really important, that what we put on our plates has an impact that goes beyond the plate. So I think that's really, really important. And then we want to teach our students, how can we make better foods, more sustainable foods, that consumers enjoy. So that's where all the technology and it's like hard work. All the science and technology has to go into this to make this happen. And we want to teach and enable our students to go out of our program and then work in the food industry and make this happen.
It's so remarkably inspiring and energizing. I'm so excited about the true hub that is forming at UMass Amherst around both research and education. So thank you for sharing what all is on the horizon. So excited to potentially see an alt protein project chapter at UMass in the future, and would love to wrap with a question that I ask all of our speakers, which is just whether there's anything else you'd like to share with this community of scientists from across the globe, as we leave this webinar and continue working at building a more sustainable food system?
Well, I guess it's important to stay humble and keep innovating. I think that's that's the most important part.
Awesome. We will fly under that banner. Thank you so much, Lutz, for joining us today. This was fantastic. Thank you to everyone from across the globe for attending this webinar, and we hope to see you again next month for our monthly Science of Alt Protein webinar. Thank you so much, everybody. Take care, and see you again soon, Lutz. Thank you.
Thank you so much, everyone. Thanks. Bye.